My name is Ian Denton, and today we'll be talking about lipid reprogramming in cancer cells using a paper titled Lipid Metabolic Reprogramming in Cancer Cells. But first, I want to talk about lipid metabolism in general. The first little thing about lipid metabolism is that it can go through catabolism, called fatty acid oxidation. Fatty acid oxidation is just a breakdown of free fatty acids to two carbon units named acetyl-CoA, and in this process they produce reducing equivalents, which will go into the ETC to make ATP. As well, the acetyl-CoA is formed right here at the end will go through the TCA cycle to produce even more reducing equivalents, which will further go to the ETC to make ATP. Usually, this process is about a 4-step process where you have two reductions right here, a hydration, and then a lyase reaction right here. The other side of fatty acid metabolism, and most important for this lesson, is fatty acid and echinozoid synthesis. Fatty acid synthesis begins right here in the cytoplasm, where you'll have an acetyl-CoA, and it's going to be activated with a bicarbonate to form malonyl-CoA. These are the units that are going to be added onto the fatty acid chain to build it. But first, you must prime the fatty acid synthase enzyme metabolon with an acetyl-CoA, and then continue to add these malonyl-CoAs through this process. It usually involves one condensation reaction, which you'll see right here, 2 reduction reactions, and then as well as a dehydration reaction. Furthermore, these fatty acids will produce ultimately palmitate. These palmitates can go through a modification in the endoplasmic reticulum, where they will be either elongated or desaturated to linoleate. Now, this is mainly in plants. Animals like us cannot synthesize linoleate or alpha linoleate, so we must eat it. But with linoleate, this leads us to even more desaturation and elongations to arachidonic acid. Now, arachidonic acid is stored in the plasma membranes of phospholipids at carbon C2, and arachidonic acids can make many echinozoids through 2 pathways. You have your linear pathway, which makes your leukotrienes through lipooxygenases, or you have your eoxins that are made through cytochrome P450. Now, these are hydroxylation reactions right here, which is just adding oxygen onto the carbon skeleton, as well as just oxygenase reactions adding oxygen. The other pathway, which is going to be of importance, is the cyclic pathway. Now, this makes namely your prostaglandins, thromboxans, resolvins, and lipoxins. And these are ring structures that have oxygen built into or on the ring. Now, why is this important? Well, today's lesson is about cancer cells, as the title suggested. And in cancer cells, fatty acids are very important. Tumors in cancer cells are characterized by their large uptake of glucose and glutamine, and breakdown of these products yield metabolites yield products essential to fatty acid metabolism. And so most cancer cells and tumor cells grow in the same region as adipocytes, which we see right here. This allows for the attainment of free fatty acids from the hydrolysis of the triacylglycerols in the adipocytes. Again, this satisfies the strong need for the fatty acids that the cancer cells have. First is the fatty acid oxidation in the cancer cells. The cancer cells obtain these free fatty acids through the adipocytes, which we see right here, and they use these fatty acids for energy through beta oxidation, we just described. Now, we see in cancer cells that there's an upregulation of the committed step, the CAT transporter, or the carnitine acetyltransferase enzyme. Now, this helps fatty acids to go into the mitochondria and be hydrolyzed. And so this produces enough power to lead to cell proliferation or cell growth. As well is the upregulation of enzymes that are in fatty acid synthesis. This includes the fatty acid synthase metabolome as a whole, acetyl-CoA carboxylase, and ATP citrate lyase. It's usually seen that if there's a higher expression of these key enzymes, there's a poor prognosis for cancer patients. And many membranes of our tumor cells or cancer cells are found to be enriched with saturated and unsaturated fatty acids. However, even if there is a strong need for the lipids and fatty acids in cancer cells, the concentrations are kept at controlled amounts, because too much or too little could lead to endoplasmic reticulum stress, also known as ERS, and this can later lead to cell death. This becomes important later on in the slides. Furthermore, we have echinozoid synthesis, which we'll see with these right here. These are your prostaglandins. Like I said, they become important. So cancer and tumor growth is promoted by the synthesis of the echinozoid prostaglandins, and these are formed by your cyclooxygenase enzymes, known as COX 1 or 2, namely number 2. So prostaglandins in the cancer and tumor cells work to upregulate communication between cancer cells and stromal/normal cells, like right here. These are your stromal/normal cells, in relation to obtaining needed resources for growth. The use of the prostaglandins also avoids the immune response by suppressing immune cells. As well, prostaglandins work to activate cell growth, migration, and neovascularization of cancer cells, mainly in breast tumors, and they help advance the invasiveness of cancer cells by activating fibroblasts to produce a tryptophan catabolite known as kynurenine. Knowing this, we know how to treat cancer cells or know of some cancer treatments now. However, most of these treatments are undergoing tests, but they're showing promising achievements. The first of which are the fatty acid oxidation drugs. So, drugs by the name of etomoxor or ranelazine are being used to inhibit the committed step of beta oxidation, which is the the carnitine acetyltransferase. These drugs will act in the same way that malonyl-CoA would and would block the CAT enzyme and prevent the movement of fatty acids in or out. This results in a buildup of the palmitate fatty acids in the cytoplasm. Not only does this prevent an essential energy source from being used and assisting proliferation, it also results in ERS, endoplasmic reticulum stress-induced apoptosis, which is cell death. The accumulation of palmitate, which is a saturated fatty acid, alters the lipid homeostasis of the cell and begins to put stress on the endoplasmic reticulum in modifying and elongating this large accumulation of fatty acid. The stress results in a UPR signal that, if persistent enough, will lead to cell death. Furthermore, we have the fatty acid synthesis, which is seen right here. Some drugs are being used in preclinical studies that are also being focused on inhibiting the key enzymes of the fatty acid synthesis. This is namely the fatty acid synthase metabolome, the ACC, acetylcarboxylase, or the ATP citrate lyase enzyme. Inhibition of these enzymes prevents the synthesis of a key resource, the fatty acid, for the cancer cells, leading to perturbations in the lipid content as well, which can also call ERS, similar to the inhibition of fatty acid oxidation. The cause of ERS is namely through the inhibition of fatty acid synthase as a whole, as its function is completely wiped out by drugs such as orlistate or C75. This can lead to apoptosis of the cancer cell. However, many of these drugs have substantial side effects and are continuing to be studied and monitored. Now we're going to get to echinozoid synthesis. So echinozoid synthesis is mainly going to be focusing on prostaglandins, which we see right here. And a main inhibitor of this synthesis are known as NSAIDs, which are your non-steroidal anti-inflammatory drugs. And the one being used in the study in the paper was known as celecoxib. This drug is a key inhibitor of the COX-2 enzyme, which plays a major role in forming the prostaglandins used as signaling molecules or cell invasion molecules. This inhibitor reversibly binds to the COX-2 enzyme and prevents the cycloogenase activity of it, which we see right here, where the initial addition of the O2 molecules onto the ring. This is the first step in formation of prostaglandins, and this causes the reduction of prostaglandins in the cancer cells as well as slowed formation of them. This disruption has shown to decrease the cell invasiveness of cancer cells and help the cell invasiveness of cancer cells that is being helped by prostaglandins, as well as the cell growth and of the tumor and cancer cells. This leads us to our first quiz question. In about 3 to 5 sentences, I want you to explain what an echinozoid is used by cancer cells and how the cancer cell uses it. As well, in the same sentences or the same paragraph, I want you to describe the treatment option that is being used to fight the echinozoid. Now, the second part of cancer cells, and one of the most important metabolites to use, is cholesterol. Cholesterol is a very important metabolite, but it's very notably known as dangerous for blood pressure or heart disease, heart attacks, and everything. But it's actually needed for membrane structure and is a precursor to other important metabolites like our bile acids, which emulsify our fats in our intestines and stomach, as well as it's needed for steroids, which are signaling molecules. Now, there's 4 broad steps to the construction of cholesterol, and it begins with 2 carbon acetates being combined to form malavolinate, and this is our key step. This is our most regulatory step. We'll get to that in just a few seconds. The second step is just conversion of mevalonate to activated isoprenes. The third step is combining these activated isoprenes to squalene, and then just converting your squalene through to cholesterol. So in cancer cells, the high need for cholesterol is also a hallmark of tumor and cancer cells, and the use of cholesterols in such cells provides the cancer cells with growth, survival, proliferation, and everything else. And there's also, as said earlier with the fatty acids, there's an upregulation of cholesterol synthesis and the enzymes. The two main ways that the cancer cells use the cholesterol is through its participation in lipid rafts as well as independent signaling molecules. So lipid rafts are essentially just microdomains in the plasma membrane that are characterized by higher concentrations of cholesterol and various types of lipids. And the concentrations of cholesterol and lipids are kept controlled as well in these things. And so in cancer cells, lipid rafts are seen to be enhanced as well as the cholesterol has become the main component. So the poor prognosis of cancer patients are if they have a high cancer cell lipid raft count as well as a high cholesterol count. The knowledge of how the lipid raft works can serve as a future treatment area against cells, as lipid rafts and cholesterol work as to promote cell signaling, migration, invasion, and survival. As well as cholesterol as a signaling molecule. So in cancer cells, the cholesterol is kept at a high but controlled concentration because of its role in signaling and growth. Too much or too little could lead to perturbations in cholesterol homeostasis, eventually leading to ERS, which is the same thing that fatty acids did. So cholesterols can be modified and converted to other molecules that act as signals to promote cell growth and angiogenesis. This leads to proliferation and migration. This is done by making sure cholesterol is constantly synthesized at both the enzyme level, which is at your HMG-CoA reductase, the regulated step of cholesterol synthesis, and most notably at the transcription level. This involves the use of site 1 and site 2 proteases, cleaving what is known as SREBP, which is known as sterol regulatory element binding protein. This protein is in the Golgi complex, and once cleaved, it is activated and goes to activate the gene that produces more HMG-CoA reductase. This provides more enzymes to convert HMG-CoA to mevalonate, which is the committed step of cholesterol synthesis. So like fatty acid synthesis, knowing how the cancer cells use cholesterol can help us find treatments to better it. So first, we're going to talk about the inhibition of HMG-CoA, from acetyl-CoA to mevalonate. So as said earlier, the key and committed step to cholesterol synthesis is through the enzyme HMG-CoA reductase to form mevalonate. Using statin drugs like simvastatin, this key step in forming the mevalonate is inhibited by blocking the enzyme from reacting with HMG-CoA. Another way to decrease the synthesis and cholesterol concentration is by inhibiting an enzyme in step 4 of the synthesis, which is this step right here, squalene to cholesterol. Inhibition of oxidosqualene cyclase will completely block the conversion of this squalene molecule to mature cholesterol molecules. Both uses of inhibition lead to limited cholesterol synthesis and lowered concentrations of it in cancer cells. Due to low concentrations, it can no longer serve its function as the main component in lipid rafts, leading to dysfunction in the said microdomains. Without properly functioning lipid rafts, cancer cells begin to struggle in proliferation, signaling, angiogenesis, etc., leading to programmed apoptosis of the cancer cell. Furthermore, as we mentioned with the SREBP, this is a way that you can upregulate HMG-CoA, but since we know this, we can also block this way of upregulation to prevent cholesterol synthesis. So by using a drug called nalfinavir, the site 1 and site 2 proteases right here, their active site of cleaving these sections of the protein of SREBP will be blocked. This leads to an immature form and a non-active form of SREBP being produced. Because of this, it cannot bind to the gene in the DNA that relates to HMG-CoA reductase, and no HMG-CoA reductase is made to provide cholesterol synthesis, therefore lowering cholesterol synthesis and cholesterol concentrations. Due to such a large perturbation in the cholesterol concentration homeostasis, ERS is induced similarly as the fatty acid oxidation or synthesis did as well, and this persistent ERS, due to constant alterations in the cholesterol homeostasis, leads to apoptosis of the cancer cell if it cannot fix the inhibition. So our second question is, in 3 to 5 sentences, explain how cholesterol is used in cancer cells and what different courses of treatment can be taken to inhibit the cell's use of the metabolite. So in summary, our cancer cells use fatty acid oxidation, synthesis, and econyzoid synthesis to produce products and energy needed for cell proliferation/growth, immunosuppression, and cell invasion. Knowing this, we can target the key enzymes of these pathways, and cancer cells can be treated through inhibition of these enzymes that induce ERS or cell death. Cancer cells also use cholesterol as a main component of lipid rafts and various cell signaling. This allows the cancer cells to, again, promote proliferation, angiogenesis, and intercellular communication. And because we know this, we can target key enzymes of the cholesterol synthesis at either the enzyme or transcriptional level that can lead to decreased cholesterol. This results in defects in cancer cell signaling, growth, inducement of ERS, and eventually cell death. Again, this is lipid reprogramming in cancer cells. I'm Ian Denton. Thank you very much.